Chapter 3. Energy Lock-in. Organisms break down and process the energy they consume, converting some of it into easily usable substances to sustain life activities. However, energy intake has both positive and negative effects. While energy is necessary for life, it also exposes organisms to competition. Organisms face constant concerns regarding energy locus competition and predation threats. Competitors aim to occupy the same energy locus and ingestion strategy as the organism, while predators aim to intake or steal the energy that the organism has already transformed and stored. Therefore, organisms naturally intake energy while attempting to lock in as much of their intaken energy as possible. Organisms in energy lock-in struggle in dynamic environments where energy is constantly absorbed and consumed, especially in harsh and ecologically reorganized habitats with low diversity. Organisms must adopt both offensive and defensive mechanisms to strike a balance in energy lock-in. Organisms need to develop a comprehensive set of protection, interception, storage, and repair mechanisms to defend against threats from all directions throughout evolution. The first priority is to establish multi-layered physical and chemical defenses inside and outside the organism's borders. In addition, proactive mission execution systems are necessary to frequently check for holes in energy lock-in and prevent infighting, turncoats, and accidents. Protection and interception. The initial stage of energy lock-in seeks to prevent the extinction of all energy. Animals developed several organs and strategies to protect themselves against predators. These organs or strategies can be broadly classified as follows. A. Hard shells. For example, turtles and sea turtles have hard shells that help protect their bodies from natural predators. B. Spikes serve as a defensive mechanism in some animals, like pufferfish and porcupines. They are erected with the expansion of the body to scare off natural enemies when danger is present. C. Hard scales. For example, crocodiles and pangolins possess such hard scales, and they can protect themselves from predators. D. Horns are another feature in some animals that serve as a defensive mechanism. For instance, animals like buffalo and deer employ sharp horns on their heads to fend off predators and to safeguard themselves from potential dangers. E. Poison, as demonstrated by the arrow poison frog, which secretes venom as a defense mechanism to temporarily incapacitate any natural predators. F. Self-injury. For instance, to avoid the attacks of natural predators, geckos will detach their tails, and octopuses will sever their tentacles. G. Another defense mechanism is playing dead. For instance, when in danger, many insects assume a stiff, death-like posture. H. Camouflage is another effective defense mechanism. For instance, chameleons can change their body color to blend in with their surroundings, and the body colors of praying mantises and stick insects allow them to expertly blend into their environment, making it difficult for predators to detect them. Plants also have numerous protection mechanisms against harm or loss of living space, as follow. A1 mechanism is leaf drop. Numerous plants endure harsh conditions, including extreme cold and heat. To survive such conditions, many plants shed their leaves to conserve water and protect themselves. Sycamore, neem, and sequoia plants all discard their leaves to shield themselves from damage in freezing temperatures. B. Some plants develop thorns on their stems and leaves to prevent them from being harmed by animals. Long thorns can also prevent other plants from growing in close proximity and allow for better growth. Examples of such plants are roses, prickly amaranth, and cacti. C. Certain plants like eucalyptus, oleander, and tomato emit distinct scents during their growth, which prevents animals and microorganisms and deters them from growing in their surroundings. The second step in energy lock-in is to establish physical boundaries that properly isolate the inside and outside of the body. At the body level, the skin and its derived structures, the chitinous exoskeleton and the mucous membranes of the inner walls of the organs, among others, serve as boundaries. At the cellular level, there are cell walls, cell membranes, protein inclusions, and more. The skin serves as a boundary between the internal and external environment, protecting against external factors and serving as a first line of defense. The skin has the following energetic locking functions. A. Protection. It safeguards the internal organs from potential mechanical damage, pathogens, or excesses in energy levels. B. Temperature regulation. This organ enhances blood flow and heat dissipation, reducing blood flow to maintain body temperature by constricting blood vessels. C. Evaporation control. The skin functions as a semi-permeable barrier that reduces the evaporation of body fluids, remaining relatively dry. D. Water resistant. The outer layer of the skin, known as epidermis, guards the subcutaneous oils from being harmed by external moisture and inhibits the loss of nutritious energy from the body. The cell walls of plants, fungi, algae, and prokaryotes are both rigid and flexible, offering structural support, shelter to cells, and preventing the intake of poisonous substances. The plant cell wall comprises 3 layers: the primary wall, the secondary wall, and the intercellular layer that separates the two. All plant cells possess primary walls on both sides of the intercellular layer. The secondary wall is situated inside the primary wall. It is formed through a secretion process after the cell ceases its growth cycle. This secretion thickens and strengthens the cell wall, while preventing direct attacks from polysaccharide-degrading enzymes and pathogens. Animal cells' membrane serves a similar purpose of safeguarding the intracellular energy and filtering out toxic elements. The use of only the skin, exoskeleton, and mucous membranes as barriers is insufficient to preserve the energy balance of a multicellular organism. In response to the aggressive looting of energy by pathogens, plants and animals have developed 2 energy lock-in mechanisms as secondary and tertiary lines of defense, in addition to their primary physical defense. The primary mechanism is a second line of defense executed by conventional forces of the immune system. When pathogens penetrate the first line of defense, traditional blood forces, including phagocytes and natural killer cells, act right away. These forces emerge from capillaries at the site of the attack, engulf the pathogens that cross the first line of defense, and release bactericidal substances to eliminate these pathogens. The secondary mechanism is the production of specialized forces in the immune system that perform complex tasks, mainly composed of T and B lymphocytes. When pathogens break through the first and second lines of defense, T-B lymphocytes, the elite of the special forces, move and emerge from small high endothelial veins into the main battlefield, lymph nodes near the infection site. Simultaneously, dendritic and macrophage cells of the immune system capture pathogens, penetrate their disguise, and transfer their unique identity information to lymphocytes using antibodies. Lymphocytes can also evolve into various T or plasma cells, providing them with a more potent weaponry. Lymphocytes can identify and kill pathogens directly, as well as remove damaged host cells. If lymphocytes cannot directly kill pathogens, they produce inactivating antigens and release cytokines to attract more immune cells with superior phagocytic capability to eliminate all pathogens. Organisms with symbiotic systems, particularly multicellular organisms, are intrinsically intertwined organic structures. The natural symbiotic mechanisms in the body necessitate the cooperation between the master cell and the symbiotic microorganisms to manage them. Thus, symbiotic bacteria are obliged to protect the overall safety of the organism and work in harmony with immune cells to form a common defense mechanism against harmful pathogens. For instance, inside the human body, a symbiotic virus known as a phage can penetrate the bacteria, commandeer its physiology, and replicate into numerous phage copies. As the bacteria perish, they burst, disintegrate, and release hundreds of phage copies from the deceased bacteria, which subsequently hijack and reproduce with the harmful bacteria. Numerous phages in our nasal cavities are dedicated killers of airborne harmful bacteria in the inhaled air. Symbiotic bacteria play a critical role in the body's immune system by expressing sugar molecules, glycans, on their surfaces, which are involved in triggering the body's alarm and intelligence functions. The immune system recognizes these glycans and produces naturally occurring circulating antibodies at high levels. Certain commensal bacteria produce a protein complex called the inflammasome. When invaded by the influenza virus, they faithfully trigger the alarm, setting off a series of immune responses related to inflammation and resistance to infection in a cascading multi-step downstream response. Biological organisms utilize a highly efficient energy lock-in mechanism to boost the immune system's potency by raising body temperature. When an organism is infected by a pathogen, dendritic cells or macrophages of the immune system recognize certain features on the pathogen's surface, which prompts the release of cellular thermogenic factors. Under the influence of thermogenic factors, many excitatory substances are released by the body's thermoregulatory center, leading to the excitation of the sympathetic nervous system, which in turn triggers a series of complex physiological stress responses in the body. Fat burning to generate heat, constriction of blood vessels to avoid heat loss, and involuntary contraction of muscle cells all contribute to the local or generalized production of heat in the body. A recent study confirmed that this mechanism is effective, and that fever helps immune cells to adhere efficiently and migrate quickly to secondary lymphoid organs or sites of inflammation. The thermogenic mechanism's heat production might have a function in pathogen elimination, and its energy integrates with the body's, enhancing immune cells' reactivity and efficiency. As a distinctive organism lacking a cellular structure, viruses need to parasitize other living cells, and when they leave, they no longer exhibit any biological interactions. This lifestyle of viruses, which can freely switch between biological and non-biological forms, effectively locks up the energy of their genetic material inside. Despite having a simple structure, they still possess mechanisms for energy protection and interception. Apart from the protein shell, also known as the capsid, animal viruses like influenza viruses possess a viral envelope structure that encases the capsid externally. Derived from the host cell membrane, which carries host phospholipids and proteins, the viral envelope consists of the virus's glycoproteins. The surface glycoproteins of a normal cell envelope recognize and activate receptors on the host cell membrane. The viral envelope aids the virus in evading the host's immune system. Once inside the cell, the viral envelope fuses with the host cell membrane, hindering recognition and attack by immune cells, and facilitating the entry and infection of the host cell by the viral capsid and genome. Storage and restoration. Living organisms store energy in two common ways: short-term storage and long-term storage. These two ways are like the way memory sticks and hard disks work in computers, or like carry-on ammunition and arsenal replenishment in the military. Short-term storage serves an immediate, rapid, short-term emergency function, whereas long-term storage is characterized by large reserves that continuously replenish the depletion of short-term storage. Living organisms utilize ATP, which can be thought of as energy currency, as a short-term storage medium. However, long-term storage varies among species due to evolutionary strategies that require a balance between storage efficiency and replenishment rate. ATP is a substance that is universal to all cellular organisms, widely acknowledged in the scientific community. All cellular organisms, from bacteria, fungi, and algae to large mammals, inherit a legacy that involves the use of ATP as a medium for temporary energy storage and direct energy delivery, and varies widely in the animal kingdom. Living cellular organisms use ATP as a direct energy source because it is readily synthesized through the acquisition of extracellular materials, providing them an abundant quantity of energy for a variety of cellular processes. Additionally, billions of years of environmental influences have not significantly altered the mechanisms of ATP synthesis and energy supply, revealing that the Earth's structure and form of energy have remained largely unchanged. Adenosine triphosphate, ATP, is a highly stable and efficient temporary energy storage medium, utilized to circulate throughout the body of an organism, serving as a currency. ATP's function is determined by its structure, and its high-energy phosphate bond provides a high-energy release ratio of J per mole. Furthermore, the rapid hydrolysis and charging rate of ATP make it ideal for cellular organisms since it allows for quick energy conversion. Mitochondria and chloroplasts supply cells with ATP for direct energy transfer. ATP releases energy when the cell requires it, hydrolyzing and breaking high-energy bonds while removing phosphoryl groups. Energy released by the process is utilized for diverse cellular activities, such as cell growth, synthesis of substances during cell division, maintenance of body temperature, transportation of substances, light and electrical energy conversion, and muscle contraction. The chemical energy of an ATP molecule is typically between 100 and 150 kilojoules per mole and is stored in a high-energy phosphate bond. The human body requires 100 to 150 moles, or 50 to 75 kilograms of ATP, to be hydrolyzed for daily energy. This means that every day, the body has to hydrolyze an amount of ATP equivalent to its weight. In comparison, the total amount of ATP present in the body is only about 0.1 moles or 51 grams. Therefore, each ATP molecule needs to be reused between 1,000 and 1,500 times per day. ATP is not a storage medium and must be utilized shortly after synthesis. Long-term energy storage in cells occurs as carbohydrates, sugars, etc., lipids, proteins, and water molecules. Sugars, lipids, and proteins all require conversion to ATP in order to serve as energy sources for the body. Glycogen is a polysaccharide formed by combining glucose molecules through the dehydration synthesis process, and it serves as a form of stored energy. Glycogen's primary biological function in animals and fungi is to act as an energy store. In humans, liver and muscle cells produce and store glycogen as a secondary source of long-term energy. It provides 4.1 kilocalories per gram of energy per unit weight. The human body mainly stores glycogen in the liver, muscle, and red blood cells, with a total amount of about 200 to 500 grams, which is equivalent to 800 to 2,000 calories. If not replenished with external energy, glycogen stored in the body will deplete within 18 hours. Besides glycogen, lipids are a well-known form of energy storage. Most vertebrates have adipose tissue, a unique structure that prevents them from dying due to energy depletion, even if they don't eat for some time. Lipid molecules such as triglycerides in animals and oilseeds are the efficient means of storing energy. Lipids have a standard energy supply of about 9.3 kilocalories per gram, which is more than twice that of glycogen and protein. However, the storage volume of lipids is much smaller compared to glycogen starch. Therefore, lipids have relatively high storage efficiency. Nutrients that are meant for long-term storage in organisms are usually consumed in a sequential manner, with sugars being consumed prior to lipids. Proteins have various functions, including catalytic functions, e.g., proteases, transport functions, e.g., transmembrane channel proteins, and communication functions, e.g., signal receptors. Proteins serve as the primary carriers of life activities rather than professional energy stores, and the energy they store is mainly for self-use. Moreover, protein energy supply is inefficient, so in normal circumstances, proteins do not provide energy for ATP synthesis, only during severe starvation. Starch, an alternative carbohydrate form, has been picked as the long-term energy storage medium for plants instead of glycogen. This decision was made due to the structure of starch, which forms more compact starch grains with fewer branches. Another reason could be the high conversion efficiency of glycogen into glucose, which is swift. Bursting energy is required only by animals when hunting or escaping. Plants do not need it. Therefore, it is safer to opt for a more compact and storage-effective storage form. Water molecules both inside and outside the cell function as carriers of stored energy. Water, in addition to storing heat and maintaining standard body temperature, is also a means for energy decomposition and triggers various chemical reactions. For maintaining the stability of the three-dimensional spatial structure of biological macromolecules and membranes, it is completely necessary to have sufficient water content in the organism. Completely dried lysozyme is inactive. Its activity can only be restored when the water content per gram of protein reaches a minimum bound water content of 0.2 grams. If there is an imbalance in the nutritive energy structure of the body fluids, or an excess of toxic energy, the water inside the body can turn into a garbage bag. Eventually, the body will expel this water, along with other energies that it cannot utilize or that exist in excess. These energies comprise neutral energies that the body can't use at all, toxic energies produced as part of the transformation process, as well as high nutrient energies that are underutilized and low nutrient energies. The body no longer needs them and eventually eliminates them as excreta. Besides fulfilling the requirements of life's activities, the organism uses a portion of the stored energy to renew and repair cells. In the film Wolverine, the hero's genes enable complete repair of any injured part in minutes, which is admirable. Will such abilities exist in real life? Certainly not. Super strong and super fast regeneration and repairability surpass the limits of energy absorption, renewal, and repair of highly differentiated human cells. The mechanism that locks energy in organisms does facilitate fulfilling their energy requirements and maintaining energy reserves for life activities. Nevertheless, the three defense mechanisms cannot entirely mitigate the opposing effects of environmental energies, competition for the same energy source, and predation by natural enemies. Electromagnetic waves, particle radiation, climate, extreme weather, pollution, diseases, predation and group competition, among other uncontrollable energy sources, continuously and to different extents, erode or impair the energy in the cells of the organism, which causes cellular aging and death to become unavoidable. Naturally, no organism will remain passive in this situation, and under the assumption of energy efficiency and effectiveness, it will try to repair or replace any damage promptly. The cycle of cell replacement varies among species. For instance, in the human body, skin cells renew approximately every 28 days, liver cells renew every 180 days, red blood cells renew every 120 days, and bone cells can take as long as 7 years to regenerate. Roughly every year, the body replaces 98% of its cells. The rate at which cells renew within an organism should correspond to the level of tissue differentiation. Due to tissue cells being scarcely differentiated, certain mollusks possess superior regenerative and healing capabilities than humans. When a hydra's head is severed, it will regenerate another. Despite being cut into several small pieces, each section of the hydra can grow into a fully functional creature. Multicellular organisms' channels for water and nutrient transport, such as blood vessels and ducts, can undergo quick repair and regeneration because of their low-level differentiation during evolution. After a doctor sutures a ruptured blood vessel, new tissues and cells grow from the suture and restore the vessel. Nerves can also be silently repaired and rejuvenated, despite being less adept at self-repair and regeneration than blood vessels. However, repairing internal organs may require specific conditions. For instance, in the liver, the remaining hepatocytes can regenerate along the reticular scaffolding, even after the surgical removal of the diseased lobule, as long as the scaffolding remains intact, resulting in the recovery of the lobule to its original state. Stem cells play a primary role in repairing the body. They belong to an unspecialized and primitive cell class that can regenerate different tissues and organs without full differentiation. Multicellular tissues contain stem cells that can divide into specialized cells through mitosis and differentiation. Additionally, stem cells can also self-renew to provide more stem cells. Adult stem cells comprise bone marrow stem cells, cord blood stem cells, and peripheral blood stem cells. They reside in specific adult body tissues. Progenitor cells can originate from them, which differentiate into functionally specific stem cells such as bone marrow MSCs, blood stem cells, and neural stem cells. Adult stem cells can generate new stem cells or differentiate into new functional cells under specific conditions and according to a particular program, effectively upholding a dynamic equilibrium between tissue and organ growth and aging. Adult stem cells play a crucial role in the regeneration and repair of various tissues and organs, including the epidermis and hematopoietic system in adult animals. The autophagy mechanism is the alternative cellular repair method discovered by Japanese scientist Yoshinori Osumi. Autophagy involves cellular cleaning and recycling of metabolic waste products. In times of stress, such as starvation, autophagy plays a critical role by quickly providing energy via direct food supply. After a cell is infected, cellular autophagy eliminates foreign intracellular bacterial and live viruses. Furthermore, the cellular autophagy mechanism supplies the necessary components for repairing and renewing damaged proteins and organelles.